Right. For this week, we're going to see reproduction and embryonic development. And in particular, we're going to see how embryonic tissue develops and how the organs we are familiar with in the past couple of chapters are developing. Let's see. Human embryonic development. All the events that occur from the time of fertilization until the animal is fully formed, we call it embryonic development. Embryonic development is any development that occurs between the time of fertilization to the end of the first 2 months. We call that embryonic development. And from the 3rd month till the point of birth, we call that fetus or fetal development. So embryo and fetus are not the same thing. Embryo is the name we give for the unborn baby before 2 months, actually within 2 months, and when you say fetus, it includes the time from 3 months to the point of birth. The developing embryo has a lot of membranes surrounding it, and we call those extraembryonic membranes. And these membranes are really important for protecting and nourishing the embryo. Some of the organisms that have these extraembryonic membranes include the mammals like us and reptiles and birds. So these are the extraembryonic membranes I told you about. We have the chorion, we have the yolk sac, we have the allantois, and then we have the amnion. Allantois, okay. So the chorion is the one that's continuous with the placenta. In fact, it is part of the placenta. And it is through the chorion, and which is also part of the placenta, that nutrient exchange takes place, nutrient and waste materials. Nutrients from the mother to the baby, and waste is from the baby to the mother. And the yolk sac, these are the sites of our blood cells. This is the site of the first blood cell formation. The blood cells we know, the red, white, and the platelets. And the allantois these are the blood vessels. These are the blood cells, and these are going to be later, they are going to be the blood vessels of the umbilical cord that takes in blood and waste materials from the fetus to the mother and vice versa. The amnion contains the amniotic fluid, this whole fluid, which is light blue color. And it is covered by the amniotic sac, and it cushions and protects the embryo. So when you talk about embryonic development and fetal development, usually we are talking about sexual reproduction, which results from fertilization. So fertilization results in zygote, and zygote comes from when the sperm and the secondary oocyte are united, that creates fertilization. And as you remember from the last week, the plasma membrane of a secondary oocyte is surrounded by the zona pellucida, which is the outer cover of the ovum from last week. And this zona pellucida is covered by follicular cells, which we call them granulosa cells. When it comes to sperm has acrosomal enzymes, and these acrosomal enzymes digest the zona pellucida so that the genetic material from the sperm can be deposited into the ovum. Sperm binds to the oocyte plasma membrane, and then the sperm nucleus enters. This leads to secondary oocyte to complete meiosis II and become an egg. Okay. And then the egg and sperm nuclei fuse and zygote is formed. Changes in zona pellucida prevent binding and penetration of additional sperm, which we call polyspermy. That's why at any given moment and under normal circumstances, it is only one sperm that penetrates egg to form just a single zygote. This is a close-up of how the sperm penetrates the egg. This is the sperm. This is sperm head, and you can see there's the nucleus. There is the acrosome, and there are these follicular cells, which we call them granulosa cells. And then acrosomal enzymes, they digest the zona pellucida and the plasma membrane. And then the genetic material will be sent toward the nucleus. So we're going to have a fusion of those two nucleuses, creating a diploid nuclei, which is called the zygote. Early embryonic development. This one is when you say embryonic period, we are actually talking about the first 2 months. That means the first 60 days. So 6 days of development, the embryonic development, occur in ovida before embryo implants in the uterus. Following fertilization, the embryo undergoes cleavage, which is cell division without growth. We're going to see this one in a minute. And this results in a tightly packed balls of cells. Yeah, we're going to see this one in a minute too. We have a picture that clearly explains this too. And the morula continues to divide and hollow out to form a blastocyst. Let me jump in a second. That's way. Okay. As you can see, there was one fertilized. There was ovulation and then fertilization took place because there is this one sperm that fertilized the egg, and then we have zygote, which divides into two. So this is the 2-cell stage, 4-cell stage, and 8-cell stage. Here's a point that I want you to pay attention to. And imagine this one is some kind of cylinder. And as you can see, the size of the cylinder is the same both on the 2-cell, 4-cell, and 8-cell stage. So technically, this whole number of cells still have the same size. Their size didn't change. And as you can see, there are 2 cells. When they divide, they are 4. So the size of the cells is gradually decreasing because to maintain the whole morula, this is, by the way, the morula. This is also the morula. It's just this one is tightly packed, and we prefer the 16 or the 32 cells as morula. But the point is, these cells are getting successively smaller. You see, this was big, then smaller, smaller, and even smaller. Because the number of the cells is increasing, but at the same time, their size is decreasing. So cleavage is a process where the number of cells is increasing while their size is decreasing, and the whole, how could I say, the whole size of those cells is actually maintained to a certain level. So this is what cleavage is: cell division without growth. And this results in a tightly packed ball of cells called morula. The morula continues to divide and hollow out to form a blastocyst. And a blastocyst is going to have, let's say this is the blastocyst. Like that. This is the blastocyst. And blastocyst has cells that are on the outside, and there are other cells that are on the inside. So, and there is also a fluid filled cavity. So this one is the blastocele, which is the fluid filled cavity. And it has an inner cell mass, which becomes the embryo. These are the shaded area, that's the embryo, the inner cell mass. And has an outer cell mass, which becomes the placenta. So this outer membrane is going to be the placenta. Blastocele, the embryo, and the outer layer is the placenta. When the embryo implants, this placenta, the outer layer, forms and secretes the HCG hormone. This is how we test for pregnancy. And it has also another function, which is maintaining the corpus luteum. When you maintain the corpus luteum, usually ovulation and menstruation don't happen. So that's what SCG is. Clear or drawing. So this is the process: ovulation, fertilization, cleavage, and then morula, which is a tight pack. There is no empty space within these tightly packed cells. That's why we call it morula. And then we have the early blastocyst. I'm going to maximize it a little. See these outer layers, we call them the prechorion, which are going to be part of the placenta. And this inner cell mass, this is going to be the embryo. And this whole thing is called blastocyst, blastocele. And then implantation occurs later. The latter embryonic development is gastrulation, or it's characterized by the formation of gastrula. Gastrula occurs during the 3rd week of development. Still, we are under embryonic development, which is within the first 60 days. And it is evident when certain cells invaginate at the primitive strake, creating 3 layers. So I'm going to redraw this one. So this is blastocyst. And we have the inner cell mass, like this one. So this was the embryo. Now what's going to happen is some of the cells from the embryo are going to get inward. That's what this invaginate means. So certain cells are going to invaginate toward the inner part of the embryo. So at the end, you are going to have a cell mass that looks like this one. So it has an outer mass and has an inner mass, and in between we have another group of cells who are going to forgive my drawing. Okay, so we have outer, middle, and inner cell mass, and these form the 3 embryonic layers, or we call them the germ layers. And the amnion surrounds the embryo. So this embryo is actually surrounded by the amnion with its own fluid. Okay. So gastrulation is all about the formation of these 3 layers, or we call them embryonic germ layers. We have the ectoderm, mesoderm, and endoderm. And from these tissues, actually, if you saw the title on the syllabus, it says tissue development and then organ development. When you say tissue development, we are talking about these 3 layers: ectoderm, mesoderm, and endoderm. We are going to see what each layer is going to develop into, and it is from these tissue layers that organs develop. So these are the tissue layers or the embryonic germ layers. We have the ectoderm, which is the blue cells, including this one, the mesoderm, the pink one, and the endoderm, the yellow one. The ectoderm, these include the nervous system. That means the peripheral and the central nervous system. It includes the epidermis of the skin. And we have the epithelial lining of the oral cavity and rectum. These are, in a sense, they are exposed to the external environment. For example, our brain is not like the heart. It's actually on the outside compared to the heart. The heart is really deep down in our abdominal cavity. So These are, in a sense, they are on the outside. And the same thing for oral cavity, it interacts with the external environment and rectum too. Mesoderm is the section that develops into muscles and skeleton or bones, and it develops into connective tissue. This one develops into nerve tissue and a little bit of epithelial tissue. This one develops into muscle tissue and connective tissue, right? The dermis of the skin, the cardiovascular system, because the heart is a muscle, and blood vessels are also composed of muscles, smooth muscles usually. The same thing for urinary system, it has muscles. Reproductive system, it has muscles, for example, the uterine wall. And the outer layers of the respiratory and digestive system. Pay attention here. It is not the inner, it is the outer, because the outer layers usually have muscles. The inner layers are made of epithelial tissue, and those are either ectoderm, like in oral cavity and rectum, or they are endoderm. So endoderm will have the epithelial lining. You see the names epithelial here, and there is epithelial here. So in terms of tissue makeup, ectoderm could be a nervous tissue or epithelial tissue. Mesoderm is purely muscle tissue and connective tissue. And endoderm could be epithelial tissue, like the lining of the digestive tract and respiratory tract, and the glands, as well as the urinary bladder. So these are actually entirely epithelial tissues. These are entirely muscle or connective tissue, and this one is nerve tissue and epithelial tissue. So this neural tube and this one is the outer layer of the ectoderm. It's going to be part of the nervous system, and we have the somites that are going to develop into muscle and bones, and there is also the notochord here. Okay, and this is going to be part of the gut. The gut is the opening that starts from the mouth and ends in the anus. That's called the gut. Neurulation is related to the nerve system. So this signals the beginning of organ formation. So the first organs to form are from the central nervous system, especially the brain and spinal cord. The newly formed mesoderm cells coalesce to form the notochord. When we say newly formed mesoderm cells coalesce or support each other or unite to form notochord, we're talking about this one, this notochord. See, they are pink, they are somite, but they are separate from the rest of them. And then neurulation is all about neural plate development. So neural plate is going to have a lot of folds, and these folds become the neural tube. So neurulation by itself is actually the formation of the neural tube, which actually pinches off from the ectoderm. It comes from here, as you can see, this blue. And the anterior portion becomes the brain, and the posterior portion becomes the spinal cord. Okay, I'm gonna annotate here. So in a sense, the development of the brain and spinal cord looks like number 9. So the anterior portion becomes the brain, and the posterior portion becomes the spinal cord. This is the anterior part, this is the posterior part. Induction is when one tissue or organ influences the development of another. We call that induction, because the development of the spinal cord is signaling the development, the development of the brain is signaling the development of the spinal cord. So the development of the brain actually helps in the development of the spinal cord. Gastrulation and neurulation, midline mesoderm, that didn't contribute to the notochord, now becomes the two masses of tissue. Okay. So what it's saying is these pink cells are the somites, right? So these somite cells that do not contribute to the notochord, or those cells that didn't become this notochord, are going to develop into muscles and bones. That's what it means. This whole thing is going to develop into muscle and bones. In particular, some of the bones are the vertebrae and the muscles are obviously everywhere. So we have the vertebrae and muscles of the axial skeleton. They develop from those somites. Neural crust consists of, lateral to somites, mesoderm cells split to form a lining of xylem. And neural crust consists of a band of cells that develops where the neural tube pinched off from the ectoderm. Cells migrate to various locations where they contribute to the formation of skin and muscles, in addition to the adrenal medulla and the ganglia of the peripheral nervous system. Okay, what it is trying to say is this whole section is developed into the muscles and bones of our body. Especially, for example, for muscles, you can take adrenal medulla. It has a muscular part and then it has glandular part. So this part is going to actually develop into a lot of things. By the end of third week, already dozen of somites are evident, and the blood vessels and guts have begun to develop. Okay, so blood vessels are derived from the somites or the mesoderm, and gut develops from the endoderm. So at this time, we actually see a lot of developments. Organ formation continues. Human embryo at 5 weeks has limb buds that are going to develop into the legs and the hands. Arms and legs develop from limb buds, and the head enlarges and sense organs become more prominent. The umbilical cord is going to develop, which connects the embryo with placenta. And as we have said earlier, allantois is going to be the blood vessel. Yes. And the yolk sac is going to be the blood cell right here. Okay, from the 6th to 8th week, there will be a remarkable change, and the shape of the fetus looks like the shape of the embryo. Actually, it looks like a human. Reflex reactions start at this stage, including the startle response. So organ formation continues. So what we're saying here is in the first 2 months or in the first 60 days, all the tissue and organ development is, organ formation is complete. The formation, tissue and organ formation is complete. And after the 2nd month, it is actually development that's going to start. It is like increasing in size. The foundation is done in the first 2 months. After that, for the next 7 months, all that's going to happen is just growth and development. It's just a matter of size and shape. So this is how a human embryo looks like, probably in two months. So we have the heart, we have the liver, lymph, which is going to develop into a hand. We have the digestive tract. We have the umbilical vessel that connects, that takes in nutrients and gives off waste materials to the mother. This is the eye. This is the somite that develops into the umbilical, the vertebrae and muscles. Placenta, the fetal side is contributed by the chorion, and the maternal side consists of uterine tissue. And there are chorionic villi. These are kind of microvilli, and these are surrounded by maternal tissue. The whole purpose of villi is to absorb nutrients from the mother and give off wastes to the mother. This is all about for exchange. Carbon dioxide and wastes move to the maternal blood, whereas nutrients and oxygen move to the fetal blood. Harmful chemicals can cross the placenta. This is actually something that happens usually. This happens after 2 months. Harmful chemicals are less likely, I'm not saying 100%, but they are less likely to affect the embryo than the fetus. The fetus is highly susceptible. The placenta is more permeable when the fetus is growing than when it was at just an embryonic stage. So this is how the embryo looks like. We have the extra embryonic cavity. This one, we have the amnion with the amniotic sac with light blue color. This is the fluid, the amniotic fluid. We have the umbilical cord that has umbilical artery and umbilical vein. We have the endometrium, which is the uterine wall. We have the developing placenta. As you can see, this pink color is continuous up to here related to the chorion, because chorion is actually part of it. It's continuous with placenta, except that this chorion is on the fetal side. And then we have the villi, the micro, they are like microvilli, except we call them chorionic villi. These are really important for absorption of nutrients. And these are maternal blood vessels. Regarding fetal growth and development, fetal development takes place between 3rd and 9th month. Embryonic development happens in the first 2 months. And there is an extreme increase in size, in particular weight and length. Genitalia appears at this stage. And the targets of beauty salon, I call them, hair, eyebrows, eyelashes, fingernails, toenails, these actually appear at this stage on the 3rd month. And from the 5th to 7 months, fine downy hair covers limbs and trunk, and later they disappear. There are things we get when we are as fetus and things we lose after birth. So these hairs are going to be lost. This is how a 6-week fetus looks like, and this is how a 12 to 16-week fetus looks like. This means this is from 3rd to 4th month, and this one is just an embryo. We can hear fetal heartbeat at 10 weeks, which is 2 months and 2 months and a half. Okay, I'm going to talk about the vernix caseosa. Okay, as you can see, there is amniotic fluid, and here is the fetus. So this amniotic fluid might damage the skin. That's why there is an outer covering of this fetus that protects the embryo from the amniotic fluid. So that's what the vernix caseosa means. It protects the skin from the amniotic fluid. And a baby which is 22 weeks old has a chance of survival if they get a proper care. And then the stages of birth. When the fetal brain is sufficiently mature, then the hypothalamus causes the pituitary to stimulate the adrenal cortex. And then this adrenal cortex helps us to secrete androgens. So fetal brain is mature, and then the hypothalamus is going to trigger the pituitary to stimulate the the adrenal cortex, and then adrenal cortex is going to produce androgens. And the placenta uses these androgens to make estrogen. And the production of estrogen is good for the production of oxytocin and prostaglandin. And as you know, oxytocin is really important for uterine contraction and a final expulsion of the fetus. So it has a lot of steps. Brain is mature, and the hypothalamus affects pituitary, pituitary affects adrenal cortex leads to the production of androgens, and placenta uses this one to make estrogen, and the production of estrogen helps to activate the production of oxytocin and prostaglandin, and that leads to uterine contraction and final expulsion of the fetus. These are the 3 stages. First, there will be rupturing of the amniotic sac because of the dilation of the cervix. This is first stage. The second one is the baby is born and the umbilical cord is cut. But still there is placenta inside, and when that placenta is delivered, that becomes the final stage. Another name for birth is parturition. Yeah, that's all. I will probably post a couple of videos to give you a more detailed view of the tissue and organ development, but this is all I have for slides. Thank you.